sorry about that. Yes, so we'll talk about mitochondria today, and then we'll talk about chromatin and gene organization, stem cells, and cell reprogramming. And I have to tell you that those are one of my favorite topics to talk about, and I hope you will be excited as well. And then we'll finish with signal transduction, 2 lectures on signal transduction. So total of 7 very exciting, very interesting lectures. And I want to remind everybody to kind of read the textbook material before each lecture. This will help you greatly in understanding the material and memorizing all of the necessary details, and it will save you a huge amount of time in preparation for the exam. So I really encourage you to read the textbook if you're not doing this already. I want to say a few words about the format of this lecture, even though it's a huge class, lots of people, that's great, but I'd like to really keep it informal as much as possible. So please feel free, if you have any questions, comments, you want to share your ideas, raise your hand, let me know, share with everybody, we can have a great discussion going on. And so I do lecture podcasts, so like the same way like Dr. Cordula Schultz did. So you will see on the ELC there will be a file with the slides and recording of my voice. What else? I want to emphasize the active learning. So I want to encourage your participation and engagement in the class, in the learning material, because instructors found that this is a very effective way of learning, and it will have a long-term effect. So you will learn something and then you will gain the skills. So you won't forget very easily, and then you can apply those skills in your future career. So emphasis on active learning, this is the big aspect of this class. So for my lectures, I will be giving a slide. For each lecture that we'll have, there will be a slide with a list of learning objectives for each lecture. So I really encourage you to look up those learning objectives, and the goal of those is to guide your learning and kind of emphasize and highlight the most important things that will be discussed during this particular lecture. And I especially encourage you to look up those learning objectives when you're preparing for the exam. So it should be very helpful. It should organize your learning. So I will try to also give 3 pop-up quizzes, very similar format to previous instructors, so they should be very easy. No worries. And of course we'll have the breakout sessions as well, and I will try to participate, be there, and talk about the problem sets with you during the breakout sessions. But as a reminder, next week we do not have breakout sessions because it's an exam, so take this time, prepare for your exam. So today we're gonna talk about the structure and function of mitochondria. And mitochondria are remarkable organelles, and probably when you hear the word mitochondria, you think cellular energy, and you are absolutely right. So mitochondria are the power plants. They produce most of the cell energy, and they also play many other important roles in order to keep our cells healthy and functional. So today we'll talk about the structure and function of those organelles. So this is, as I mentioned, this is the set of learning objectives, and you can go over those points at the end of the lecture or some other time. So before I talk about the structure and function of those organelles, I really want to give you a broad view and broad perspective, and we'll talk about origin of mitochondria from the evolutionary standpoint. So what you see here is the graph showing a major evolutionary events in the evolution of higher eukaryotic organisms and humans. So here is the time scale. today is the present day, and here we have like 5 billion years ago, when there was a formation of our Earth. And what you can clearly see here, there are two different blocks. So the first couple of billion years, our Earth was very primitive and life was very simple, and there was no oxygen. So this blue area highlights the rise of the oxygen in the Earth atmosphere, but for the first few billions of years, there was no oxygen. So there were some primitive microbes, bacteria, that were able to survive by performing anaerobic oxidation. But this form of generation of the energy is very inefficient. And during this time, there was a very important point where there was an evolution of tiny little microbes who were able to take the carbon dioxide from the atmosphere, take the water molecule, and use the solar energy and perform a process of photosynthesis and generate the glucose and release the oxygen. And this was a very important point, because from this point on, there was an accumulation of the oxygen in the Earth atmosphere, and this totally changed the history of the Earth. So this was a very important point. But at the beginning, the oxygen was highly toxic to those organisms which were using anaerobic oxidation to support life. So those tiny microbes and bacteria had to evolve different strategies and different systems in order to survive, in order to make more energy, and in order to deal with the toxic levels of oxygen. So of course, bacteria are very small and tiny, and they proliferate very fast. That means that they can accumulate some mutations, and they can evolve systems very rapidly. So if there is an environmental pressure to evolve some system that will be advantageous, they will do it, because they are small, tiny, they proliferate very fast. And at this point, there was an evolution of aerobic bacteria who were able to take advantage of this oxygen. So they evolved set of proteins, enzymes, and in their membranes, which were able to take the oxygen and the carbohydrate molecules and perform aerobic respiration. And this was a huge advantage. Those organisms allow them to proliferate, to expand, and this was a marking point for the evolution of higher eukaryotes, because oxidative or aerobic respiration is producing much more energy as compared to anaerobic respiration. So this is the point where, about 1.6 billion years ago, it's thought that this is the point of evolution of the mitochondria. And how it happened. So there is an endosymbiont theory. This is a widely accepted theory explaining how the evolution of mitochondria and also chloroplasts happened. So here we have an ancestral eukaryotic cell. So this cell has a nucleus, and this cell is probably using anaerobic oxidation, so not able to use oxygen efficiently and not able to produce much energy. But in the primitive Earth, when levels of oxygen were rising, this cell had to deal with that. And it had two options: either die from rising and toxic oxygen levels, or acquire a system which would allow to deal with the oxygen, to make more energy, to detoxify cells from the oxygen. And so this theory says that this ancestral primitive eukaryotic cell was able to accomplish this goal by acquiring a microbe. So the bacteria who was able to do the aerobic oxidation, use oxygen efficiently to produce energy and support life. In summary, this endosymbiont theory says that mitochondria and chloroplasts evolved from bacteria, which were acquired through those ancestral eukaryotic cells. And this acquisition process involved endocytosis of bacterium. So here we have two bacteria cells, or maybe, so this is the bacterium that was acquired inside the cell, and then over thousands of years it became a permanent component or organelle within this cell. So there was this symbiotic relationship between the bacteria and the primitive eukaryotic cell. And this event gave rise to the evolution of higher eukaryotes. So this is, like I said, this is a widely accepted theory of the origin of mitochondria, and we have plenty evidence for this. So what kind of evidence we have? So when you study mitochondria, you immediately see a remarkable, striking similarity between mitochondria and bacteria. So mitochondria have a double membrane, just like bacteria. When you look at the shape and size of those organelles, there is a striking resemblance between the mitochondrion and bacteria cells in the same shape and size. Mitochondria have their own DNA, so they have their own genome. They also have their own ribosomes, and both the DNA structure and organization is very similar in mitochondria and in bacteria cells. And mitochondria reproduce by fission, and the word reproduce is not maybe the best, I should say divide, or they can split by the fission process, which is highly similar to bacteria reproduction. So bacteria cell will grow, divide its DNA, and then from one cell you will have two cells. And bacteria can do very similar process, so they can undergo fission or division in a very similar way as bacteria cells. So what's the advantage of having mitochondria? So as I said, a formation and acquisition of mitochondria was a prerequisite for evolution of complex animals, and this was for purely economical reasons. When you look at the amount of energy, so the highly organized organisms, highly complex organisms, require a lot of energy in order to compete, in order to adapt to different environments. And so when we compare anaerobic glycolysis with aerobic oxidation, from one molecule of glucose, we can produce only 2 molecules of ATP energy when the organism is doing anaerobic glycolysis, and 36 molecules of ATP through aerobic oxidation. So this is 15 times more energy. And everybody needs more energy in order to evolve, become bigger, more competitive. So this aerobic oxidation performed by mitochondria was a key in the evolution of higher eukaryotes and us humans. So at this point, I want to play a YouTube movie, and this will be a really nice refresher and reminder, a little bit maybe from your biochemistry course. And I really like this movie because it shows in a very elegant way how mitochondria produces the ATP energy. There's no sound.